Good day, ladies and gentlemen. Welcome to Immunology and Immunobiology. This is part of the list of courses you are going to take under MSc Medical Microbiology program. For those of you that were new here in the Department of Microbiology, I welcome you once again. And for our students that they have been here since undergraduate, you know how we are operating here in the Department of Microbiology. Today, in the beginning of this course, we are going to focus on the concept of immunology and structure of antigen. Immunology, as you know, is just the study of immune cells and how they are operating, their category, their structure, their interaction, their strength of association, and also their kinetics. Whereas immunology is a study of the physiology and anatomy the immune organs, cells, and molecules. As simple as that. There are a lot of other mechanisms that are taking place within the immune system and are responsible for maintaining the security and defense of a human body. We are going to see the relationship between antigens and antibody. As you know, these antigens they code across bacteria, viruses, fungi, parasites, and other complex molecules in form of toxins and poisons. There are also self-antigens and other alloantigens. They are also playing a very vital role in the organs of the immune system. One of the most important organs of the immune system is a bone marrow. Bone marrow, you can find them in the bone of the ribs, in the bone of the femur, which is the longest bone in the human body, and it is responsible in hematopoiesis, which is the production of blood. This production of blood can be white blood cell, red blood cell, but the whole of the cells of the immune system they comes from the white blood cell. So the most important cells here are the B cell and the T cell. The bone marrow produces the B cell and also the T cell, but most importantly, the B cell they are maturated in the bone marrow, whereas the T cell are maturated in the thymus. Thymus is located just behind the sternum, which is the central bone of the chest, very close to the heart. While the T cell are producing the bone marrow, they are maturated in the thymus, and that's where the name came from, which is thymus standing for T cell. We also have the spleen. The spleen is located in the upper abdomen and is responsible for blood filter and sorting of the immune cells. Whereas the lymph node is not a localized organ. You can find it in so many parts of the body, including the neck, the armpit, and the groin, and it is functioning in immune filter and serve as a checkpoint for antigens or microorganisms when they enter the human body. We have cells of the immune system. The first one are lymphocytes. Lymphocytes are B cells and T cells. As you know, even the B cell and T cell, they also differentiated into other subset. And we also have macrophages, neutrophils, dendritic cells, we have mast cells, and we have a lot of other cells. All these cells are working together with the organs of the immune system concurrently to ensure that they defend the body. We have also the molecules of the immune system. The antibodies are also called the immunoglobulins. These antibodies, they are Y in shape and they are located on the surface of the B cell and they function in neutralization of antigen, opsonization as antigen presentation using these macrophages and major histocompatibility complex. They also work in agglutination, which can be visibly seen during blood grouping and other in vitro antigen antibody tests. They also function in complement fixation and antitoxin action. Another molecule of the immune system are the cytokines. These include the chemokines and the interleukins. The chemokines and the interleukins, they work based on a chemical gradient. They work as a means of communication where if they increase a certain concentration of the chemokine or the interleukins, then they can attract the function of the B cell in that location. That's why I say they are working using chemical gradient. They can work either through autocrine, paracrine, and endocrine. Then we have also the complement fixation. This enhances the pathogen destruction via also opsonization, chemotaxis, and the action of lysozyme, which eventually cause apoptosis and necrosis of a cell, be it either via classical, alternative, and lactin pathway. There are uh, three categories of immunity. These include innate and adaptive immunity, which is natural and just like a cell-mediated immunity. Then there is active and passive immunity, immunity acquired via artificial inoculation, and immunity acquired via passive transfer of immune cells from mother to daughter, and humoral and cell-mediated immunity, which straightforward the main B cell and T cell. The innate immunity and adaptive immunity, they differ by the rate of specificity. The innate immunity is just a natural immunity. It is physical and is something that you can vividly see because it includes some certain organs and tissues. This include the skin, the barrier, the fluid, the enzymes in the gastrointestinal tract, the tears, the lysozyme in the tears, and other enzymes in the saliva, the sputum. So there are uh, parts of the immune system that you can see clearly, and they defend the body as a first line of defense, and they don't have memory. Whereas the adaptive immunity, they protect the body and they work very slowly because at the end they produce memory B cells, and they include B cells as antibodies and T cell. They produce memory, especially in the case of B cell after differentiation, which is very effective in ensuring that they protect the body during subsequent infection. And the active and the passive immunity. Active immunity is an immunity that you can acquire either through artificial action via vaccination or when the B cell recognize a pathogen within the body. As I've said, they can differentiate it into plasma B cell, the memory B cell. So they can restore, they can ensure effective immunity through production of memory B cells and also by acquisition of vaccine through artificial inoculation. And this is long lasting because of the memory of the B cells. Whereas the passive immunity, this is an immunity that usually a child acquires via his mother, which can be transferred through a placenta or umbilical cord and also through breast milk feeding. As well, a child can be inoculated using artificial passive immunity by vaccination, most especially uh, rabies vaccine and also snake bite vaccine. This is a short time before the child starts developing his immunity actively and artificially. Humoral immunity straightforward means immunity from T cell and B cell. In the case of humoral, it's a B lymphocyte immunity. The mechanism usually is when B cell recognize the presence of an antigen with the help of antigen presenting cell, It is held by a T cell to ensure that they transform into plasma B cell, which immediately start working, and then a memory B cell, which are stored for future intervention. So the main goal of this humoral immunity is antibodies traveling through the blood to attack pathogens for destruction, neutralization, or dump invaders together through agglutination so they can't infect the cell. Whereas the cell-mediated immunity is just a T lymphocyte, a thymus cell, and it works by first of all displaying the invader or antigen on the surface of the major histocompatibility complex and other antigen presenting cells. Then it triggers the activity of cytotoxic T cell, which is termed as the cluster of differentiation 8. This recognizes the signals and bind to the infected cell, and eventually it can use purpurins or increase uh, the volume of water within the cytoplasm, and eventually uh, the porocyte or the B cell or the antigen will burst using a process known as the apoptosis. The goal of the cell mediated is to destroy the factory entirely and to eliminate the pathogen without further replication. The structure of immunoglobulin is Y in shape. However, there are different types of immunoglobulin. We have some of them, they are monomers. We have one dimer and we have one pentamer. But what joined them together is a variable chain and the heavy chain, and together they come together using a disulfide bond. So the variable part has two heavy chains and also the light chain, two light chains. The variable part is responsible for binding with the antigen. As well, there are other parts that they play a biological function. This include the constant region below, the the binding the binding part of the antigen effector function, and also the hinge region, which is functioning as a source of flexibility for the immunoglobulin. These are the classes of the immunoglobulins, and you can use a formula called the gamete to recall them. We have IgG, IgA, IgM, IgE, and IgD. As I've said, some of them are monomers, most especially the IgG, which is abundant in the blood circulation, and also the IgE, the dimer, because it has a protruded extension, so it is diametric in nature. It is there in the saliva and the breast milk, so usually passive immunity transfers IgE to the child. And we have the phantomer, the first antibody produced during infection. It has a five extended structure of the Y-shaped immunoglobulin and is very prevalent whenever there is the presence of infection. Then we have the IgE, which is involved in allergy and parasitic infection. And we have another monomer mainly found in the B cell surface. So they are on the surface of the plasma and memory B cells, and they have an intervention of secondary infection or during secondary contact with a particular antigen. The concept and structure of antigen. Antigen is just a complex polysaccharide or protein. Its structure and the rate at which it can trigger immune response depend on its antigenicity and immunogenicity. So antigen may have a single epitope, which is a binding site, or a multiple epitope. So long as it has a complex molecular and chemical structure, it can be antigenic and immunogenic. In the left, you see that there is a binding between the antibody and the antigen, and the antibody is binded to the antigen using paratope, while the antigen is binded to the antibody using epitope. So these are receptor site, and they are playing a very vital role during antigen-antibody reaction. The properties of antigen. For an antigen to be a perfect antigen, it has to be foreign to the body. It's not safe. So the body will recognize that this is a foreign substances and may cause a problem into the system. So it can trigger an immune response. Number two, before an antigen become immunogenic and antigenic, it, it is determined by the molecular size. So the higher the molecular size, the higher the immunogenicity. The lower the molecular size, the lower the immunogenicity. But usually a molecular size of 10, 100,000 Dalton is serving as a highly immunogenic antigen, where a four antigen with 5,000 10,000 Dalton is in an antigen may be antigenetic but not immunogenetic, means it cannot trigger an immune response. Where there is such kind of condition, it is augmented with haptin, which is an antigenic but not immunogenic. So a combination of haptin with an adjuvant, which is immunogenic but not antigenic, produce a very strong antigen that is immunogenic and antigenic. Number three is the chemical complexity. The shape of the antigen and the chemical complexity also determine how strong the antigen is. Some antigen they have rough surfaces, means they have multiple epitope sites and they are highly chemically complex. So this is something that determines if the antigen is antigenetic and immunogenic. Also, an antigen is specific in action because it is only binding to the antibody via the paratope through its epitope. So therefore, its action is specific through confirmatory reaction between antibody and antigen. Also, an antigen have to be degradable. So after binding to APC, which is the antigen presenting cell, usually the body degrade it and break it into smaller pieces so that it can be removed outside the body. In a situation where an antigen is not degradable, then it is not a very good antigen. And this is done usually using by the action of macrophages and dendritic cells and other antigen presenting cells. These components of an antigen include epitope, which is the binding site, receptor site to the antibody, and we have also the autoantigens, example, the DNA and also the corneal tissues. There is also the alloantigens, which are antigens similar to the blood grouping antigen. They are self-antigen, but they can be recognized. We use this principle in determining human blood grouping because these are a common antigen that you can find in virtually every human being. Biphenophilic antigen are identical antigen found in cells of different species, carried out in individual 1 and individual 2. Then there is an adjuvant. It's also another type of antigen, but as I've said, it is not immunogenic, but it is antigenic. So they are just opposite to haptin. So the combination of adjuvant and haptin brings about a perfect antigen that is immunogenic and antigenic. Antigen and antibody react in this way, as you can see below. Most antigens are binding to the antibody in the variable part. You can see that this is a heavy chain and the Y component of the antigen. So this is the part where the antigen and the antibody are binding together. The part from the antibody is called the paratope, then the part from the antigen is called an epitope. So collectively they produce a reaction, and the reaction between one antibody and one antigen is called affinity. So in this case, it is a complex antigen antibody reaction where two antibodies are reacting with one antigen via two epitopes from the antigen. So antigen antibody interaction properties include highly specific reaction. Specificity in the sense that it is not an engulfment, it is not phagocytosis, it is a binding between the antigen binding site and the antibody binding site. So that's why it is highly specific. Whenever there's a reaction, is because the antibody is extremely specific to the antibody, to the antigen. And in the course, in a observable manner, if you take example of agglutination and precipitation, it is an antigen antibody reaction that you can see it vividly. You take example the blood grouping, you can see the agglutination. The Waddell test, you can see agglutination. The strip test for the detection of HIV and other types of infection, you can see the agglutination. That is because the lines you see between the control and the test are forming because there is a reaction between the antigen and the antibody. And the reaction is non-covalent because it is reversible. Any change in the physicochemical properties during the reaction, this includes change in temperature, change in pH, it can cause a reversible reaction to so in such a way that the antigen and the antibody can be detached again. So at a favorable condition, they can form a complex and they can be detachable. So that's why the reaction is reversible. So the reaction doesn't cause any denaturation of the antigen or the antibody, depending on the prevailing cause. To measure the strength of antigen antibody reaction, we have to consider that antigen antibody reaction, their binding is defined by three things. Affinity, the strength of the binding between one antigen and one antibody, and also avidity, the overall strength of the binding between several antibodies and several antigen. Collectively, they produce a complex of antigen antibody, but what matters is the valency. How many antibodies are binding to how many antigen? So the valency matters. The higher the concentration of the antigen should be equivalent to the higher the concentration of the antibody. So the valency can determine the strength of binding between the complex of the antigen antibody reaction. Another thing that can also determine the strength of the binding in the complex is the structural arrangement of the epitope on the surface of the antigen. Some epitopes may have several binding sites, or some may have one binding site. An antigen with several binding sites can be binded to different antibodies. So even from there, you can understand that the valency is not the same. So the strength of the complex may be weaker. That's why these things matters. The valency and the structure of the epitope as they bind through epitope and paratope. And there is a cross reactivity. It refers to the ability of the antibody to bind to similar epitope of different antigen. As I've explained from here you see one antigen is binding to two antibodies because the antigen have two epitope site. So this is a cross reactivity, and it also determines the affinity and the avidity of the binding between the antigen and the antibody. There is a kinetic, the rate at which this antigen and antibody are binding to each other. How rapid, how quick. So how rapid and quick antigen antibody bind to each other is determined by the prevailing condition. At optimum temperature, there will be a rapid binding, and if the valency is the same as well, the pH also matters. So at the optimum condition, there is a rapid combination between the antigen and antibody, and the kinetic of the avidity and the avidity can be measured uh, using equilibrium of binding between the antigen and antibody, which we are going to discuss in the next topic. And as we have explained, it is reversible. It is a situation where the complex can be reversed based on the prevailing condition. Then there is a dependency concentration, as I've explained, which is the valency rate. The higher the concentration, the higher the rate of binding, which is similar to law of mass and action. The rate of chemical reaction, as you know, is directly proportional to the concentration of the reactant. So if the number of the antigen is high, as well the number of the antibody is high, then the rate at which it forms a complex will be rapid. So we use kinetics to know how strong is the binding and how fast is the binding, how fast is dissociation and association between the antigen and antibody, considering the prevailing condition at the time of the reaction between the antigen and the antibody. And these are the conditions. So there is temperature. At optimum temperature, there will be effective reaction between the antigen and antibody as well. If the pH is optimum, usually 6.5 to 8.5, there will be a very good reaction and it will be rapid. Then there's also ionic strength concentration. The kind of solution we use to monitor the antigen antibody reaction also matters. Usually we are using saline solution, so the higher the concentration of the sodium chloride will establish a gradient so it can slow the reaction between the antigen and the antibody. So it have to maintain an optimum concentration to ensure effective binding, effective association and dissociation of antigen. Then the availability of protein at the moment of the reaction. Take example, proteolytic enzyme are used to enhance the antigen antibody reaction. However, the concentration will also be optimum to ensure effective binding. Number five is also talking about the balancing, the concentration of both the antigen and antibody during the reaction. And there is also the antigen binding site. So how many epitopes are available at the surface of the antigen and how many antibody can bind to a particular antigen, provided it has either a single epitope or multiple epitope. And there is structural arrangement. Structural arrangement, how many binding site and how many antigen can bind to specific antibodies available during the reaction. Stages of antigen-antibody reaction. We have primary stage, which is rapid and reversible because the antigen and antibody are binding for the first time. And during the second stage, it is slow but is very visible with either in vitro or in vivo. In a situation where it is inside the human body, it's not something that can be visualized, but there are preconditioning signs and symptoms that we can use to measure the reaction. In vitro outside the body is something very straightforward that we can see. Agglutination and precipitation is clear in the case of certain assays. Take example, block grouping, cross matching, and other type of tests is observable, is something that we can see vividly during the reaction. As well, complement fixation can also be noticed, but not in vivo. There's also neutralization by increasing number of antibodies prevent the proliferation of the antigen. And there is immobilization to stagnate the antigen at the location where it get access to the human body. Then there is opsonization, antigen presentation by manifesting the epitope on the surface of the antigen after phagocytosis. So thank you for listening. The link here is a quiz link. You can follow them, you answer the question within 5 minutes, and this will serve as your attendance. So make sure that you do the needful, and we shall see in the next topic. Not that immediately after the Salah festivity, we are going to resume a physical class. We are just doing this online class to make sure that we cover some certain part of the course before we return. It's very essential that MSc is conducted via physical interaction between the lecturer and the student. So we are just doing this to cover the time before the main contact when we return. Thank you very